Stanford University.
Good afternoon, everyone. I'm Natalia Kuri. I'm a lecturer in the Department of Bioengineering, and I'm also Director of Educational Programs of the newly established UCSF Stanford Center of Excellence in Regulatory Science and Innovation. And so we are very excited and proud to present our first speaker here at Stanford today, Dr. Carolyn Wilson. And she's the Associate Director for Research at the Center of Biologics Evaluation and Research. And so the title of her presentation will be very fitting to our first lecture, and it's going to be about Introduction to Regulatory Science at the FDA. So welcome, Carolyn.
Thank you.
We're very happy to have you here.
Okay. Well, thank you for having me. I'm glad that this has worked out. And I have been the Associate Director for Research at CBER for about 8 years. And before that, I was what's called a researcher reviewer within CBER. And I'll not talk a lot about that, but because I'll be explaining what that means as I get through my slides. I am happy to be interrupted because I have put together a very ambitious slide deck, and I'm probably going to whiz through a lot of it because what I've done is I've organized the talk around a strategic plan that the FDA put out about 3 years ago that identified 8 major priorities for our regulatory science program. And what I find is that regulatory science is one of those terms that people use, but they don't really understand what it means. And the best way I have found for people to really understand what it means from our perspective as a regulatory agency is to provide some examples. And so I'll be doing a lot of running through examples. And so if I go through some really fast, just for the sake of time, but it's actually something you're interested in, then stop me. I can slow down and do that. Yes.
Is your deck posted?
I don't know.
The whole presentation will be publicly available after the lecture, but the slide decks and the whole thing.
That was the other reason I thought just leaving all the slides in there, even if I skip over a few, that people can look at the examples. I don't go through quickly. Then I go through too quickly. All right. Oh, all right. Yep. Sorry, I have this thing here, so that's why I keep not knowing what to do with my arm. All right. So what I'll be talking about is how the FDA uses regulatory science to really fulfill our mission. And our mission is very broad. And, I have to do the disclaimer first. My comments are an informal communication, represent my own best judgment, don't bind or obligate FDA. But a lot of people don't really realize the very broad scope and responsibility that the FDA has. But the products that we regulate represent 25 cents of every GDP dollar. It represents all of the foods as well as cosmetics. We don't regulate meat, that's regulated by USDA, but all other foods are regulated by Center for Food Science and Nutrition. Center for Veterinary Medicine regulates products that are used to treat animals for veterinary medicine, with the exception of vaccines, which is done by USDA. Don't ask me how these things got distributed. And then they also do all animal feed, is also regulated by Center for Vet Medicine. And then Center for Drugs, obviously people are probably most familiar with Center for Drugs because that's what's in most of your medicine cabinets, are all the products regulated by them. But then we also have Center for Devices and Radiological Health. And this is, also a very broad remit. It's everything from, very standard, simple things to, much more complex devices. And, now that we're moving into more and more of a mobile world, there's a lot of interesting complexities. Next-gen sequencing being used for diagnostic purposes of human disease, personalized medicine, precision medicine, whatever term you like to apply to that. Those are some of the cutting edge technologies that are being seen by that center. And then the center I'm in, which is Center for Biologics, and I'll be going into a lot more detail about what we do in CBER. But we have a product portfolio with a very large public health impact, all the blood, vaccines, and so on. And then our newest addition to our center is, our agency is tobacco. And in the area of tobacco, we now have the legal authority to regulate manufacture, distribution, and provide restrictions on certain additives that might be appealing to young people, new warning boxes and so on. Some of you may have even seen the new ad campaign that the FDA has launched about, trying to dissuade young people from starting smoking. So our mission is to protect the public health, to assure safety, efficacy, and security of human and veterinary drugs, biological products, all the things I just mentioned, except this is an old mission statement because it doesn't have tobacco products. And we advance the public health by speeding innovations to make medicines more effective, safer, and more affordable, making sure that the public gets accurate science-based information to use medicine and food to improve health, and regulating manufacturing, marketing, and distribution of tobacco products, as I mentioned, especially with reduction of use by minors, and supporting the nation's counterterrorism capability. So we do all of this under a variety of different legal authorities. There are statutory authorities, which means these are laws that are enacted by Congress and signed into law by the President. And the Food, Drug, and Cosmetic Act is sort of the grandfather of them all, along with the Public Health Service Act. And then more recent iterations of some laws that provide powers to the agency are things like FDAA, FDASIA, the various user fee acts, and so on. But then we also promulgate regulations under these laws. These are really the agency's interpretation of what these laws mean, because as you can imagine, when Congress writes a law, it's still fairly broad, but we need to interpret it in a more detailed way. And that's in the Code of Federal Regulations. And most of our regulatory regulations are in Title 21. I've listed just a few examples here. And then guidance documents are a really important tool for us, because these are a way for us to represent to the industry that's being regulated our most current thinking based on the best available science. And guidance documents, unlike laws and regulations, which are requirements, guidance documents are not binding to either us or to the sponsor. So I don't mean for you to be able to read this slide, but this is really just to show, this is one example, Part 610, the General Biological Product Standards. It gives you a sense of the level of detail that's involved in a regulation. It goes through all of the various things that need to be done as far as the product and the requirements that it needs to meet, labeling standards, and so on. And so this is just, to give you a sense of the complexity. And what's challenging for us as regulators is that depending on the product, you may not actually have all of the scientific data and understanding and knowledge about a product to really address all of these aspects. And that's where regulatory science becomes a really important part. So in terms of looking at the big picture, the way the FDA approaches regulation of, for example, novel medical products is we have this umbrella of the legal framework and the regulations. But then we also, especially when you start talking about new areas of science, we need to have external discussion through advisory committees, open public workshops. We rely heavily on regulatory science, and I'm going to talk more about that for the rest of this talk. Obviously, the data that's submitted to the agency is obviously an important component of information, and then we use all of that to have internal discussion to develop rational policy and decisions. At least that's our hope. The FDA has defined regulatory science as the development and use of scientific knowledge, tools, standards, and approaches necessary for the assessment of FDA-regulated product safety, efficacy, quality, potency, and performance. Can hardly get through it without a breath. And it's a very detailed definition, but it's also, for most people, not a very meaningful definition. So what I hope to do in the rest of this time is to convey to you really what this definition means. And so one of the ways that I do that is through this graphic, which really shows sort of the major aspects of the regulatory component in terms of a new product, which is you need to be able to characterize your product, you need farm tox data, clinical protocol design and analysis, risk assessment, and then when it's licensed, post-marketing surveillance. But a lot of times, especially as you're starting this process with a novel product that's relying on new technology, you may not have all the information that you really need to be able to assess this, to make a decision about this going into humans. And so that's where regulatory challenges come up. Maybe you don't understand enough about the mechanism of action. Maybe there's not even a good animal model to be able to get proof of concept data or safety data. You need to be able to understand what are the critical attributes of the product to measure. And so this is where regulatory science, through a combination of both discovery and targeted development of new tools, can start to address some of those gaps in our knowledge that we need to be able to make these very tough regulatory decisions. And that then informs the development of policy and regulatory decision making. As we get that kind of information back out to sponsors, then the idea is that sponsors who are developing these products have better data that they can submit to the agency that's filling some of these gaps, and that we are in a place where we can really look at the benefits and the risk of a particular product, and hopefully at the end of the day, license a product that's going to have a positive impact on whatever the public health need is that gave rise to this new pathway. Some of the things that are unique to the FDA scientist who's a part of this machine is that, first of all, we know the kinds of questions that need to be answered. Secondly, because we're inside the FDA, we can see across a whole portfolio of products. So we may be able to identify gaps in knowledge that could advance a whole product class forward as opposed to an individual product. So I mean, obviously individual companies who are developing vaccine X are responsible for developing data to support the licensure of their vaccine. But if that vaccine platform is based on, a platform that could be improved, or for example, let's talk about TB, where we still don't fully understand even what are the correlates of protection for TB vaccines, and we want to develop new TB vaccines. That's the kind of work that we can do to kind of help push that whole field forward. So FDA has an intramural component and an extramural component. And most of the examples I'll be providing to you today are really from our intramural research program. The extramural research has really just been, maybe in the last 5 or 6 years, been really expanding. And we do that through a broad agency announcement that provides contracts for specific projects, and then the Centers of Excellence in Regulatory Science and Research, as Natalia mentioned. And we now have 4, one of which is UCSF and Stanford. And the CERCs are really focused on advancing the field of regulatory science and helping to, again, have more effective and efficient product development and evaluation. And in addition to that, the thing that's unique to the CERCs as opposed to, for example, the FDA intramural program is also the training and the collaborative piece, professional development and scientific exchanges. All right, so I'm going to talk a little bit more about our specific center. So you got an idea that there's a very broad remit for the entire agency. And so I'm going to focus in on Center for Biologics, and we regulate what are called the complex biological products. So most of what are now sort of traditional biotech products like monoclonal antibodies, recombinant proteins are actually regulated now in Center for Drugs. But we have still a lot of what I call the messy biological products. So they may be living cells, living viruses, like a live virus vaccine or a living virus that's adopted to be used for gene therapy. And these things raise all kinds of concerns in terms of the source materials and how do you maintain sterility when they can't be terminally sterilized. You can't do the kind of processing of these products that you would, say, a recombinant protein or monoclonal antibody. There's a lot of complexity. Allergenic products shown here represents those extracts that are used for both diagnosis and treatment, and this is actually over about 1,300 different extracts that we understand to varying levels of degree. Therapeutic probiotics is actually a fairly tame New area compared to fecal transplantation, which we also are regulating. So you can imagine the challenges of dealing with those kinds of products. And then things like xenotransplantation, cell and gene therapies, and all the blood and blood derivatives, and of course all the vaccines. So the nature of our products is that we have a very huge public health impact in terms of having a major role in disease prevention. But also when you talk about a vaccine going into basically a universally used healthy population, often infants or young children, the bar for safety is going to be very different than, say, an oncology product going into an end-stage cancer patient. And that can be a challenge in terms of how you regulate that. We do a lot of work in national preparedness. We're involved in seasonal and pandemic influenza. We actually are involved in strain selection each year. We go to the World Health Organization. We develop seed strains for each season's flu vaccine and develop reagents that we distribute to manufacturers to do quality control testing of those lots and so on. So we have a very important role for national preparedness. But a lot of these products don't necessarily have huge market incentives. It's a lot different than the blockbuster drug for treating heart disease, right? And so that is another challenge in and of itself. So a lot of our products actually have a very heavy government engagement by NIH, BARDA, and so on. And then we also have a lot of very new technologies like cell and gene therapies. And tomorrow is an all-day workshop that's being organized by Dr. Baer and his colleagues at Stanford, as well as NIST and FDA, to look at stem cell products and how do we characterize those. And I'll talk a little bit more about some of those complexities. And the challenge with new technologies, as you all know, is that everybody thinks that's going to be the next great thing. Maybe some of you even saw the 60 Minutes show last week on using poliovirus, replicating poliovirus to treat brain cancer. It sounds really exciting, but it's like monoclonal antibodies was going to be the magic bullet in the 70s, right? And then really they didn't become ready for prime time until about 20 years later. That's really hard for the public to understand why those kind of really complex, challenging technologies take so long to develop, because there's a lot of hype around them. So gene therapy is another one. I mean, that, back in the early 90s, right, that was going to treat all the metabolic disorders, and I think, we're getting closer, but we're not there yet. There have been a lot of bumps in the road in terms of gene therapy. So CBER is somewhat unique in the FDA in that we have what's called a researcher reviewer model. And what that means is that these individuals do all the same full-time review activities, going out on inspections, reviewing data that's submitted in submissions, organizing advisory committees, writing guidance documents, but they also spend up to half their time running or participating in research labs. And because of this, because they sort of wear these two different hats, it allows our staff to really be able to see what's not even hit our doors yet, what's coming out, because they're going out to the scientific meetings and they're staying up to date in their field, but also can look again in-house at this portfolio of products. And so it allows us to be both proactive and responsive, and I think that's a really important component, again, to regulatory science, especially when we have a lot of new and exciting science that's getting translated into new medical products. We need to be prepared. We can't really wait till it gets here and then start thinking about what are the issues and what are the gaps. So to try to start addressing those kinds of things, the FDA published about 3 years ago the Strategic Plan for Regulatory Science. And again, the idea is that it can really help to speed innovation and improve regulatory decision making, getting the products to people quickly, and that regulatory science can be a driving force for FDA in order to help promote this public health mission. So I'm not going to read these because I'll be going through them, but there's 8 different priority areas. And the first is modernizing toxicology to enhance product safety. And this is really a very exciting area. Maybe some of you have heard of things like organs on a chip, using microfluidic technology to make basically lungs on a chip. But on the other hand, it's also very daunting to take these really cutting-edge new approaches to doing toxicologic screens and being able to validate them and show that they really are predictive in terms of what's going to happen when you go into humans. And some of these models can also be a combination of in silico and animal data. So some studies that are being done at the National Center for Toxicological Research is to integrate what's known about human data along with animal data to decrease our uncertainty when looking at various toxicology data around oral dosing and food exposures. We also do work around very mechanistic approaches. So for example, adjuvants. There's a lot of excitement in looking at novel adjuvants. And again, this is very important in the era of pandemic influenza, because having a good adjuvant is a dose-sparing approach. If you don't have to give as much of a dose of flu vaccine, you can get it out to more people, and that is going to be more cost-effective and also faster to prepare for a pandemic. But a lot of these novel adjuvants will have local or systemic reactogenicity that's not necessarily good. And so what Hannah Golding's lab has been doing is looking at, for example, a panel of cell lines with different human toll-like receptors on them and asking which ones get activated, and then looking at things like in vitro assays in monocyte macrophage cell lines and the cytokine profile, what's pro-inflammatory versus going to be a good immune response, and then combining that finally with an in vivo immunogenicity evaluation. She has found that prostaglandin E1, for example, is a much earlier biomarker of reactogenicity than IL-1, which is, or fever, which is actually what most people use. So these are some new approaches. This doesn't quite fit in this particular priority, but it's a great example of some work that CBER scientists have been doing. This is work led by Todd Merkel, which has an amazing public health importance. So some of you may have heard about the resurgence of whooping cough in the last few years, and I believe in 2014, CDC reported something like 15,000 new cases. And yet this is in the face of pretty much universal vaccination. So what's going on? Why is that? Well, as you may know, there was a change about 20 years ago from a whole cell pertussis vaccine to an acellular pertussis vaccine, thought to be safer than the whole cell vaccine. What Todd Merkel has shown is first he developed a non-human primate model that actually recapitulates the human disease. And then using that model, he was able to show that if you vaccinate with the acellular pertussis, you get still actually fairly robust replication of the Bordetella vaccine. Bordetella bacteria, excuse me, compared to the whole cell pertussis, which gives you nearly sterilizing immunity. And in the same PNAS paper, he shows that these animals are able to transmit the bacteria and have it cause disease. So even though these animals don't get disease themselves, they're propagating, they're basically like a silent carrier like typhoid Mary. So this is very important because this provides insights into how to develop potentially a new vaccine that could address the problems that we're facing. Clinical evaluation, personalized medicine, these are really important areas because as you probably know, this is a very expensive and time-consuming part of new drug or new therapy development, is the time it takes to do all the clinical trials. So if you can do refinement and reduce that time. So one of the things that CDER scientists have done is they actually did meta-analysis of a variety of different phase 2 and 3 and pediatric studies of 5 different drugs for hepatitis C virus, and they were able to show complete concordance in looking at 12 weeks and 24 weeks. Previously, they always required 24 weeks for these studies. And so now, based on this, they have the confidence to say that you can do these studies for new antivirals for hepatitis C virus with 12 weeks of data instead of 24 weeks, which basically cuts that clinical trial development in half. product manufacturing and quality is very important, especially again in my world of biologics. And I have an interesting example that came up in the last few years with immunoglobulins, which are used for a variety of different clinical indications. And for years, actually, we had been following and observing these thrombolytic events that were associated with the use of these products, and many of them were very serious: myocardial infarction, stroke, so on. But nobody really understood why this was happening until there was a cluster of cases associated with a specific lot. And what was found by Mikhail Ovanesov is that factor 11A, which is one of the clotting factors, was found in these lots, and this was the amount of thrombin generation with a spiked factor 11A preparation, and this is one of the thrombolytic lots. So it pretty much demonstrated that that was the causative factor. So this led to a voluntary product withdrawal by the manufacturers of these products. We looked at all of the immunoglobulin products at open public workshops, and his lab has been doing transfer of this assay methodology, so the industry now has incorporated this into their lot release testing, and we now have an international reference material to facilitate industry adoption of this method to do quality control and evaluation of these products. So as I mentioned, emerging technologies is again another challenging area because you don't always have all the data you need. And I think in the area of stem cell derived products, there's a lot of challenges because a lot of times the product that you use may be different after you implant the cells. They may go different places. They may respond differently to the local milieu depending on where they go in the body. And so understanding in this very complex population of stem cells, what are the critical product attributes that correlate with a desired outcome or correlate with an undesired outcome is going to be a really important way for us to regulate this. So we have an in-house consortium that's working on multipotent stromal cells, and I'm not going to go into a lot of information on this because this will be discussed at the workshop tomorrow, but they're looking at a variety of different parameters, for example, using microarray to look at RNA expression, also single-cell RT-PCR, mass spec-based proteomics, epigenetics, and karyotyping, and then taking this array of product characteristics along with traditional flow cytometry and trying to correlate that with functional outcomes like animal models, immunosuppression, or differentiation capacity. And so one example of the work that they've done is a very quantitative assay for looking at adapogenic activity, which is one of the cell types that multipotent stromal cells differentiates into. And they have 7 different donor-derived lines. They've basically made large cell banks at passage 3, 5, and 7. And what you see here on the y-axis is the number that differentiate into adipocytes. And as you can see, you see variability between donors and over time between the different passages. And some donors, some of the cell lines don't even get past passage 3 or 5. Interestingly, several years ago, there was an International Society for Cell Therapy, and they developed a set of cell surface markers, 6 cell surface markers. So everybody agreed this is what makes us define an MSC. All of these 7 different cell lines look pretty much the same based on this ISCT panel. So it just is a nice demonstration that these cell surface markers aren't really enough to define what these cell products are. There's a lot more heterogeneity that's not captured in that particular panel. Next-gen sequencing, a lot of excitement. There's a lot of work going on in the FDA to prepare for this. One of the areas is high-performance integrated virtual environment. I've put a few references about that describes this, but this is work that's being led by Vahan Simanyan, and it's based on using an InfiniBand technology to do highly parallelized both data storage and data computation through a web-based interface. And we've developed a lot of in-house algorithms that allow us to have a very facile mix and match to address different biological questions. And also he's developed what's called a honeycomb database model, which allows for much faster implementation of new databases. And then we have a lot of visualizations associated with the different algorithms. One example of work we've been doing to apply this is in the area of looking at deep sequencing to look at vaccine consistency. So the poliovirus, the live virus polio vaccine, has an issue with neurovirulence, and well, probably all of you are too young to know this because we haven't used the live virus. well, most of the people in the room, but you'll remember the problem with neurovirulence. And we actually, for many years, had to use a monkey test to assess every single lot of poliovirus. It was actually in the regulations. And so we're now looking at, are there more modern methods to do this? And so looking at this particular mutation that's associated with neurovirulence shows that using next-gen sequencing, you can very easily pick these kinds of mutations up. Diverse data is a very important area, especially in the post-marketing area and electronic health records, health claims databases. And again, an area that's very important for us is the safety of the flu vaccine because it goes into so many people. And when the H1N1 vaccine was implemented in 2009, one of the big concerns was whether or not it would be associated with Guillain-Barré syndrome. Again, most of you won't remember this, but in the 70s there was another swine flu highly related to the H1N1 strain that was circulating in 2009, and it was associated with an increased incidence of this autoimmune chronic neurodegenerative disorder called Guillain-Barré syndrome. So obviously that was of great concern. So doing a very large meta-analysis of safety data, that we looked at 23 million vaccinated individuals from 6 different healthcare surveillance systems. And by doing this, we did find a very small statistical increased risk of Guillain-Barré syndrome per million people vaccinated, 1.6 excess. But if you compare that to the risk of just getting infected and the risk of death and hospitalization, those risks are about 500 times higher than the risk associated with vaccination. So we felt that this was very important information to reassure the public about the safety of this vaccine. Food safety is a very important area. Our products are more and more coming from all over the world. This doesn't really project very well, but I mean, if you can read it, it's basically just showing how in your salad, every single aspect of it is coming from anywhere in the world, basically. And so making sure that those products are safe. Every time there's a foodborne outbreak, I'm sure many of you have followed in the news in the recent years, you know, that by the time this product enters commerce and is contaminated with salmonella or listeria or whatever it is, using the traditional ways of getting the clinical information and doing DNA fingerprinting and so on, you know, it would take several weeks to identify the source of contamination. By that time, you know, the incidents of the foodborne outbreak are almost resolved. So we're looking very closely at the ability to use next-gen sequencing to be able to resolve these outbreaks much faster. And because if you can intervene here, you'll obviously prevent a lot more cases. And so we've been doing this. We actually have Illumina sequencers in a variety of different regional laboratories so that they can do the sequencing as they collect the samples. And we've shown that this network of sequencing is reliable, efficient, providing good specificity, and we've been able to actually use it to define foodborne outbreaks in at least 15 different examples. And actually, the most recent listeria outbreak, I think, was done through next-gen sequencing. Medical countermeasures is a very important area for the agency. The challenge in medical countermeasures is that oftentimes you can't necessarily do an efficacy trial because the infectious agent that you're trying to develop a vaccine against may not be circulating, and so it's not ethical to obviously do challenge studies. And so in that case, we've developed this new approach, which is called the animal rule, which is when you can't get human efficacy data, that you can do this in animal models. But then how do you make sure you have appropriate animal models, or how do you get studies that can be useful? So that's another area we've been working in. We've actually developed luciferase labeled anthrax, and using IVIS, which is in vitro imaging system, we can do much more precise staging of the anthrax infection by looking at sites of replication. And this is really important because a lot of the products are post-exposure prophylaxis or treatment, and so by having more accurate staging, you can get better data. The social and behavioral science is a very important piece, and I'm just have one quick example from Center for Food Science, but this is really important in the tobacco arena, drugs arena, vaccines. I'm sure you all know about, the recent measles outbreak because we have families who aren't getting their children vaccinated because they still think vaccines cause autism when that paper was retracted how many years ago, and everybody has disproven that hypothesis. But so getting the communication out clearly, effectively is a big challenge. Nutrition is obviously a really important thing. We're all dealing with the diabetes issue epidemic, and so one of the things that Sufsan looked at is, can we impact how people consume products that have 2 servings by having a different labeling approach? Yeah.
Will the FDA be also tackling this whole GMO debate?
I'm not the best person to comment on that. I think genetically modified organisms are actually, if it's animals anyway, are regulated by Center for Vet Medicine. The issue of, you're asking whether or not a product is going to be labeled as GMO. Yeah, I think again, so this is my personal opinion, not official FDA opinion, but I think a lot of that is wrapped up in a lot better consumer education too, because a lot of times just because it's genetically modified doesn't necessarily mean that there's an increased risk. It could actually even be a good thing. And so I think that's one challenge I think the communication piece is probably a very big aspect of that. But I don't know what the agency's official position is. I don't want to get it into trouble. Very quickly, like you guys, I understand you just moved into this building last year. We moved last summer. This is our new life sciences building, and we moved there last summer. That's one of our typical labs before we moved in. But we're really excited because probably like your new building, you've probably thought about it and designed it to be able to support the technology and the studies that you want to do. And so we've designed it with a lot more space to have core capacity for certain technology to help support our scientific research, expanded vivarium, very much expanded BSL-3 capacity and dedicated space for things like high-resolution NMR, mass spectrometry, microarray, PCR, and so on. We're really excited because we just are getting installed an 850 megahertz magnet in the NMR facility that's going to facilitate doing high-resolution structural information to look at, for example, biosimilars and trying to figure out how can we look at a biosimilar and show that it's really the same as the reference product. That's a very important, challenging area. I want to just mention that we don't do all of this by ourselves, that even when it's an intramural program, we heavily collaborate with a variety of different partners, especially academia as well as other government agencies. In the last few minutes, I'm actually whizzing through here. I want to just give you an example. This is from my own former life when I was a researcher reviewer, and I was in what's called the Division of Cell and Gene Therapies. And in the early to mid-90s, with the new class of immunosuppressive therapies, people thought that they could use non-human sources of tissues or organs to address the organ shortage. And pigs was one of the ones that was a source animal that a lot of people thought would work really well. But because I was trained as a retrovirologist, I was actually brought to the agency to work in the area of gene therapy and was doing research on gene therapy. But as I started to think about and see submissions coming in using pig cells for treatment of Parkinson's disease and things like that, it occurred to me that, well, especially endogenous retroviruses that are part of the germline, but other infectious agents in this context of a very heavily immunosuppressed host, chronic cell-cell contact, viruses doing what they do best, mutating, recombining, reassorting, that you potentially create an environment to select for a novel infectious agent that may cause disease in the recipient or may be transmitted to others, in which case you have a greater public health concern. So we actually took that question into the lab, and we got primary peripheral blood mononuclear cells from NIH mini pigs, and then just non-specifically activated them with PHA and PMA, and we were able to show, this is a reverse transcriptase activity, that we could transmit RT activity in either a pig or a human cell line. So it showed that you can isolate an infectious virus straight out of pig blood, basically. And we actually showed later that we could also do the same thing with plasma. We could isolate infectious virus. And so this, along with a lot of other public discussion, generated the Public Health Service Guideline on Infectious Disease Issues in Xenotransplantation, which goes much broader than just the issue with this retrovirus, and then the FDA guidance, which also has specific guidance on this pig endogenous retrovirus. But what I want to point out with all of this is that the translation of research findings into a regulatory environment doesn't always happen overnight. And so, you know, as I mentioned, in the mid-90s, we started to see more and more interest on the part of submissions coming into the agency. I started these experiments basically in '95, published a few years later. And during this period from '95 to '98, it's not like this research wasn't informing our thoughts. We were having open public meetings, our advisory subcommittee, to discuss the data and the risks and develop our own recommendations, which we were providing advice on a daily basis to sponsors about what they needed to do. But it didn't develop into actual guidance, written guidance for industry until a few years later, and then finally the final version of the guidance wasn't issued until 2003. So I think it's an important thing to remember, which is that regulatory science, while it often informs our regulatory decision making and can drive policy development, there's often a lag time. So you don't always see like an immediate one-to-one correlation. So I'll just finish by saying that the regulatory science program at FDA really helps to fill a unique niche to facilitate product development. And by also having a cadre of scientific experts who understand the regulatory process, it allows us to be able to be proactively addressing regulatory science gaps while also providing in-house ability to respond to public health and regulatory emergencies. For example, you know, H1N1 in 2008 to 9. And I'll just stop here on this last one that has a few URLs that might have additional information if you want to go a little deeper on any of this. And I'm happy to answer any questions.
For more, please visit us at stanford.edu.